All right, so we'll move into 7.2, which is going to talk a little bit about DNA replication. We'll talk about RNA. So DNA and chromosomes, this is a little bit of a review. So in prokaryotes, remember that the DNA is one circular chromosome, and there also can be many additional pieces of DNA called plasmids. In eukaryotes, the chromosomes are going to exist in many linear forms. So chromatin is the form of DNA during interphase, also known as the normal life cycle or normal life phase of the cell, when the cell is doing what it's meant to do, not replicating. And then as chromosomes, this is form of DNA during cell division. And remember that DNA is going to be bound to proteins called histones. DNA and histones units are called nucleosomes, and those nucleosomes clump together in order to form chromosomes. All this entire slide is review. All right, so structure of RNA is very similar to the structure of DNA. There's a few differences. So RNA is a nucleic acid that contains similar parts as DNA. It contains a 5-carbon sugar. Okay, so this is going to be the difference. DNA, it's deoxyribose. In RNA, it's ribo. Okay, or ribose. I'm sorry, ribose. It contains a nitrogenous base and a phosphate group. RNA and DNA are different in the following ways. RNA contains the sugar ribose instead of deoxyribose. RNA is going to be single-stranded while DNA is double-stranded. And RNA contains a nitrogenous base uracil, which replaces the nitrogenous base thymine in DNA. So it's A and U go together, G and T go together with RNA. All right, so RNA's purpose is that it serves as a temporary copy of a gene. Okay, so the DNA is the permanent blueprint, so to speak. It's the one that is kept safe. It's the one that if it gets damaged, all future plans could be in jeopardy. RNA is going to be the temporary construction site copy. So it's the photocopied version that's taken to the work site. It's okay if it gets dirty or if it's okay if it gets messed up because we have the master copy safe on lock and key. So DNA is a temporary copy. RNA is a temporary copy of the DNA instruction. So you have different types of RNA, which is going to be important as we start moving through transcription and translation. So mRNA is messenger RNA, and this is RNA that carries the information about the protein from the nucleus to the ribosomes. rRNA is known as ribosomal RNA. This is RNA that makes up the structure of the ribosome. tRNA is transfer RNA, and this is RNA that transfers the amino acids to the ribosome during protein synthesis. Remember, protein synthesis is protein production. All right, so DNA replication is a process of creating 2 duplicate strands of DNA from one original strand. This process is semi-conservative, which means it saves half. So during DNA replication, the 2 strands are split, and each one of these new strands has a new complementary strand added to it. So at the end, you have 2 new strands of DNA, each of them containing one old strand and one new strand. So that's why we call it semi-conservative. All right, so the steps of DNA replication, so you have a lot of different enzymes that come together to make this process happen. So step 1 is helicase. Remember, an enzyme is anything that ends in ASE. Typically, you can tell that's the enzyme. So helicase is going to unzip the DNA strand by separating the hydrogen bonds that connect the nitrogenous bases. So remember that A's and T's and G's and C's are held together. The rungs on the ladder are held together by hydrogen bond. Helicase is going to come through and unzip that, so to speak. It's going to break that bond. Step 2, primase is going to prime the DNA strand so that it is ready to begin the process of replication. Primase is going to provide a starting point for replication to start. Step 3, DNA polymerase is going to add the correct nucleotide to the DNA strand. For instance, A's to T's, G's to C's, so on and so forth. Step 4, ligase is going to create the sugar phosphate bonds in the backbone. So the last, I believe, yep, the last slide here, we can look at these different things. So you see helicase is right here working its way from left to right, and it's unzipping these hydrogen bonds. And then you have primase that started here, primase here is causing DNA polymerase to go through and start putting the nitrogenous bases together, right? And one thing that is not on here is ligase. Okay. If there's any questions, please let me know. Be sure to complete the form that goes with this, and we will talk to you again soon. Thank you.